Hello, everybody. Welcome to another exciting IIS-sponsored webinar. It's great to see so many people from all over the world and participating in this webinar at different time zones as well. And it's going to be an exciting webinar. So probably we should give people probably half a minute or so just to settle down, and then I'll introduce our speaker for this evening. I encourage you to listen attentively and ask questions as many as you can, and I will convey these questions at the end of the webinar. Okay. All right. Again, welcome once again, everybody. This is an exciting day, and it's really good to see so many participation from all over the world. In fact, from every corner of the world. I have a great pleasure today of introducing Donna Farber, who is the George H. Humphreys II Professor of Surgical Sciences in Surgery and Professor of Microbiology and Immunology at Columbia University. The focus of Donna's research is on adaptive immunity, particularly to virus infections and how T cells differentiate and generate long-term immunological memory in diverse tissue sites. Dr. Farber's laboratory identified subsets of tissue-resident memory T cells in the lung that mediate protective immunity to respiratory viruses, and has led an initiative in translational immunology to dissect human immune responses in tissues throughout the body in multiple mucosal and lymphoid tissues from individual organ donors of all ages. She has also made important contribution to understanding how T cells seed tissues and mediate responses to viruses during different life stages, including infancy. Dr. Farber leads the NIH/NIAID funded program grants on human immunity, antiviral responses, and is part of the Human Immunology Project Consortium, as well as the NIH/NHLBI Consortium on Human Lung Aging. In addition to the NIH, her research is supported by the Helmsley Charitable Trust and the Chan Zuckerberg Seed Network for the Human Cell Atlas. She has published over 130 publications and has served on numerous advisory committees for the NIH, American Association of Immunologists, Federation of Clinical Immunology Societies, and others. Tonight, Dr. Farber is going to talk to us about respiratory immunity and COVID-19. And I look forward to this exciting presentation. Donna, it's all yours now.
Okay. Henry, thank you. Thank you for that very nice introduction. It's a real pleasure to be here. I want to also thank the IUIS for inviting me to give this webinar, and it's really a pleasure to be here. So I want to also just say hello to everybody who's joined from all over the world, and people are writing in notes. So it's right now noon in New York City. I know it's the evening in some places, so good evening. And also good morning and good afternoon to everybody. So it's great to be here with this international community to present about what has been on the minds of everyone really has been affected by the current worldwide pandemic caused by the virus, the novel coronavirus, SARS-CoV-2, resulting in a worldwide pandemic of COVID-19, which is a disease that's caused by SARS-CoV-2, and just the recent, the updated numbers now are over 7 million globally. 2 million of those are in the US, and 400,000 cases here in the state where I live, New York. New York used to have the highest number of cases, even higher than any other country, but now Brazil and Russia are more than that. But this has affected every country in the world. In terms of what happens, what are the nature of the disease, and again, everybody's very familiar with this. This is just sort of the breakdown, the numerical breakdown based on cases in China, which is probably maybe the most that have been written up thus far, although a lot of information is coming out very rapidly on this. So that there's a certain percentage, a low percentage of people infected are asymptomatic, but this percentage may be a little underestimating it for certain age groups. Most people get what's called a mild disease, which is without pneumonia. It's a dry cough, it's a persistent fever and fatigue. Even this mild disease is actually much worse than most viral respiratory illnesses. So it's much worse than influenza. It lasts for a really long time. People have fevers for weeks and feeling fatigue and associated symptoms. So this mild disease, even though it's mild and that they're not hospitalized, is still pretty severe in itself in terms of the normal illness to respiratory viruses. About 14-15% have a severe disease, which means the oxygen saturation is lower than 93%. There's lung infiltrate and people have trouble breathing. And then 5% of patients are critical, which means they have acute respiratory distress syndrome or ARDS, shock, and this can lead also to multi-organ failure and death. So the case mortality, again, some of these frequencies are changing almost all the time, but from this particular study, it was 2.3%, which is 23 times more lethal than influenza. And again, the mortality does vary based on certain risk factors. So the risk factors for severe disease are middle or older age, greater than 50 or 60 years of old, being male, comorbidities, including heart disease, diabetes, hypertension, and less than 10% of cases are under 30 years. So really age is a real factor in this. There is, of course, no approved antiviral or vaccines yet, although there's a lot of research on it. So the treatment is supportive care. So what I wanted to talk to you about this evening is the importance of respiratory immunity and how we're studying respiratory immunity and COVID-19 disease. So we know that in most individuals, the virus, the main target of the viral infection is in the lung. It's the epithelial cells of the lung. So SARS-CoV-2 expresses on its surface a spike protein, and this spike protein binds to the cellular receptor, which is ACE2 or angiotensin converting enzyme 2, which is expressed on the surface of alveolar epithelial cells type 2 in the lung. And the virus binds to the cellular receptor and in this way initiates a productive infection in these cells. Now, this ACE2 is also expressed by a number of other cells, particularly in the gut and in other tissues. And it's known that also the virus has some clinical manifestations also in the intestine. But for the most part, this is a respiratory virus that you get from inhaling airborne particles, and it infects, the primary site is in the lung. So in terms of understanding the immune response, it's important to understand the immune response to respiratory viruses. So I'll go over that, sort of the immune response to key features of the immune response to respiratory viruses, and then the importance of the lung-resident immune response, including lung tissue resident memory cells and lung macrophages in this initial response to a respiratory virus, and then how that components of this lung-resident immune response can mediate either protection or immunopathology. So sort of what we know already from other viral models. And then I'll talk about work in my lab where we've been doing studies to monitor respiratory immunity in humans to other viruses, and then a new study and some early results from a study that we've been doing for longitudinal analysis of respiratory and systemic immunity in COVID-19 patients with severe disease. So to start off, In terms of the antiviral immune response, so most of what we know about antiviral immunity is from mouse models and also studies in humans of immunity to influenza virus. Influenza is a ubiquitous virus. Again, that affects, again, everybody in the world, and it comes back again and again. And we don't always have immunity to new strains. And influenza virus, of course, has also caused worldwide pandemics, particularly 100 years ago. And so what we know about the immune response to flu is that there's an early innate immune response here. So it occurs in stages. So you have the early innate immune response, which is mediated chiefly by the production of type 1 interferon. And this is produced by the cells that are infected, as well as innate immune cells such as macrophages and monocytes that are also in the lung. This early innate response helps to control the viral load. And so you can see the viral load peaks a few days after infection. And the innate immune response helps to control the early viral load and also promotes activation of the adaptive immune response. And that would be the T cells, CD4 and CD8 cells, as well as B cells to be activated. And these are all virus specific. So adaptive immunity, you have lymphocytes that express these antigen receptors that are specific for the virus. So you have clonal expansion of virus-specific CD8 cells. They will go to the lung, and these will act to control virus by killing the virally infected cells, and thereby controlling the spread of virus. CD4 cells also, you'll get clonal expansions. They don't expand and divide to the same extent as CD4, but you get a very robust clonal expansion of virus-specific CD4s. The CD4s have multiple functions. They're helper cells, or they're going to, in the lung, they will help promote an inflammatory response by acting to promote macrophage activation, and that will help curb the spread of virus in the tissue site. And they will also help B cells to make antibody, and that will be in the lymphoid tissue, and this antibody level also will increase. You can see that the peak of the adaptive immune response, both the peak T cell response and the peak virus-specific antibody response, is much later after viral clearance. But all of these immune reactants are absolutely necessary to control the virus, to clear the virus and control it. Now, after the virus is cleared, the numbers of these virus-specific T cells undergoes a contraction. So you don't keep that high number of these virus-specific T cells around. There is cell death, and many of these go away, and you have a contraction of numbers, but it doesn't go to the baseline. A certain proportion of virus-specific CD8 and CD4 cells persist as CD8 memory T cells or CD4 memory T cells, and antibody is also maintained in the serum. And the antibody-producing cells, the plasma cells, also are maintained in the bone marrow, where they continue to pump out antibody. It is the preformed immune reactants as part of the adaptive immune response that can mediate protective immunity. So if the virus is encountered now long after this initial encounter with the virus, you will now have both memory CD8, memory CD4, and antibody pre-existing to this virus to provide protective immunity. This doesn't always happen to every viral infection, but we know it happens with flu, and it can happen. It also happens with and it's the whole basis of vaccines. So these provide protective immunity. So, but in the case of respiratory viruses, you also have to consider the localization of where all these events are occurring. And so with the respiratory viruses, these are really occurring in the lung. And particularly for T cell responses, so for the generation of effector CD4 and CD8 cells, this is happening in the lung. So the priming of the T cells, the initial activation of naive T cells, and encountering the antigen and the co-stimulatory signal necessary for T cell activation does occur in the lymphoid tissue. But this will cause a proliferation and differentiation of T cells to effector cells, and it's the effector cells that can then leave the lymphoid tissue and migrate to the peripheral sites of infection, which would be the lung. And after the virus is cleared, as I mentioned, most of these effector cells will die, but proportions of these will go on to survive as memory T cells. And there are different types of memory T cells. There's circulating memory T cells, such as central memory T cells, which will go to the lymphoid tissue, effector memory T cells, which will go and migrate to multiple peripheral sites, and sort of the newest T cell is memory T cell that has been identified as a tissue resident memory cell. So these are non-circulating memory T cells that will be maintained in tissues such as the lungs and intestines and really all the tissues in your body. The majority of T cells in tissues are tissue resident memory cells. These are distinguished from the circulating memory cells based on phenotype. They express both CD69 and CD103. CD69 is an early activation marker, also retention marker. CD103 is an integrin that mediates adhesion and interaction to epithelial cells. They're retained in tissues, and they also have a distinct transcriptional profile. And this has been defined both for mouse and humans. So humans, these tissues all have predominant populations of tissue-resident memory cells. So what about for the lung? So we know from this is from work that's been done in my laboratory, and also it's been done in other laboratories that have also shown that tissue-resident memory cells are essential for mediating protection and actually mediate optimal protection to influenza challenge. So the tissue resident memory cells here that have CD69 and CD103 are localized around the airway. This is sort of a picture of a lung, and they're localized around the airway. They persist there long after the virus is cleared. And this is in an early experiments from a mouse transfer model. We showed that mice who had flu-specific memory cells only in the lungs, so only had the tissue resident, were fully protected from a lethal challenge compared to mice that had circulating memory cells, so memory cells from the spleen, or mice that did not have any flu-specific memory cells. So the lung TRM mediate optimal protective immunity. And in work from Stanley Perlman's lab, so Stanley Perlman has done a lot of wonderful immunology with SARS, with coronavirus, and this is to the original SARS-CoV-1. This is a SARS virus that caused the initial outbreak in 2003 in China. And what he showed is that airway memory CD4 cells mediate protection. So you can see that you have memory cells in the airway, and if you deplete them by intranasal depletion with an anti-CD4 antibody, so if you have mice with airway memory cells, they're fully protected. If you deplete them, they lose that protection. So again, showing that to a respiratory virus, those tissue resident T cells are really vitally important, and even more important than even the circulating. And in fact, the circulating isn't necessarily an indicator of whether you're going to get protection in the lung. And another kind of important aspect of this tissue resident memory and sort of this tissue resident is that, in fact, if you want to follow the dynamics of the respiratory immune response or immune response to a respiratory virus, you simply need to look in the tissue. That's where all of the action is happening. And this is, again, previous work in my laboratory where we used a technique of in vivo antibody labeling. This is a technique that's been used by a number of investigators, including Dave Masovitz has done a nice analysis showing how this works in multiple tissues, where what happens is you infuse a fluorescently labeled anti-T cell or anti-immune cell antibody into mice. And in this case, we infused it into naive mice or mice that had been infected with influenza following different days of infection with influenza. And then you immediately perfuse the lungs and harvest the tissue after 5 to 10 minutes. And what happens is all the T cells that are accessible to circulation, so those T cells in the vasculature, will pick up the label of the antibody and become fluorescently labeled. Whereas those that are in the tissue and not accessible, so not circulating, will be protected from in vivo antibody labeling. And what you can do here, and this is with CD4 cells, if you look at the in vivo labeling, which would be on this part of the flow cytometry plot, and then you come back, you counterstain with a non-overlapping anti-CD4 antibody, what you can see is in the naive or mock-infected lung, most of the T cells, even from perfused lung, are labeled with the antibody because there's a lot of vasculature in the lung. And there's very low percentage that are protected. However, 10 days after influenza infection, you can see, I'll tell you that there's an influx of T cells into the lung, so you get a greatly increased number of T cells in the lung. And now the majority of these T cells are in the tissue. They're protected from in vivo antibody labeling. And even after the virus is cleared, these protected cells, you can see you maintain a significant fraction of T cells in the lung are in the tissue in that protected niche. And of those protected cells, if you look at the phenotype, these protected cells have the phenotype. They're CD69 positive. They're also CD11A high for CD4 cells of tissue-resident memory cells, whereas the label cells don't have this phenotype of tissue-resident memory cells. And moreover, the cells that are in the tissue niche are enriched for influenza specificity. So this is where your antigen-specific, virus-specific T cells are. They're enriched in that tissue niche. They're in the niche where they need to be, whereas the labeled really don't have that many. Another aspect of these tissue cells is that you can treat the mouse with depleting antibodies, so that you can deplete out circulating T cells, or you can limit the egress of cells to the lung. So in this case, we treated the mouse with FTY720. This is a sphingosine 1-phosphate receptor agonist. So it causes downregulation of the sphingosine 1-phosphate receptor, and therefore T cells can no longer leave and enter circulation and leave the lymphoid tissue. So what happens is you get a depletion of T cells from the circulation, and you get a reduction in the ability of T cells to enter, to egress from lymph nodes. And what you can see is you get rid of the circulating T cells in the lung, but in fact you maintain those tissue resident memory. And we showed previously, and others have shown this as well, is that you get that retention. You keep the same numbers of tissue resident memory. And then you can show that you can challenge a mouse that has TRM, lung TRM, with a heterotypic strain of flu in the presence. So you're blocking all the circulating responses, but just having the TRM is enough to give you that protective response. This is a primary, so this is when you don't have TRM, and this is when you do, and you can see those lung TRM mediate that protection. So again, in several ways, you can see that the lung TRM mediate optimal protective responses. So in addition, though, to protection, to a virus infection, there's also the other side of the immune response, where the immune response, in being exuberant and trying to protect and clear that virus as well as efficiently as it can, you can get a little bit of collateral damage, and that's called immunopathology. So it's well known, at least to influenza and other viruses as well, is that you can get immunopathology in the lung, and that's usually due to two specific mechanisms. One is that there could be a cytokine storm, or what's called cytokine release syndrome, and that's usually due from the innate immune system, and it tends to be very rapid. So this is just data from a study that John Taharo did when he was in Michael Oldstone's lab, which shows in the lung, in severe influenza infection, that you can get the induction, very rapid induction in the lung of type 1 interferon, as well as other chemokines and cytokines such as IL-6. So IL-6 is a key cytokine that's associated with cytokine release syndrome. And this can happen very rapidly to a severe influenza infection. And so there are certain inhibitors that can target these pathways and can cause dampening of that. So it's important to identify immunopathology so that you can identify a treatment strategy to try and dampen the overexuberant nature of the immune response that's leading to immunopathology, but still maintain protection. And then the other type of immunopathology can be T cell mediated, because T cells will cause tissue damage, because especially cytotoxic T cells. So CD8 T cells will kill a virally infected cell, so you're killing an epithelial cell. There's a lot of repair mechanisms so that damage is limited, is very limited, and the lung does repair itself. But again, if that T cell response is too overexuberant in either CD4 or CD8, because CD4 can also promote inflammation, then you will get a lot of inflammation. And this is in a mouse model that we had set up a while ago, that with memory responses that are a lot of memory T cells. These are both circulating and tissue resident, that if you have a lot of circulating memory responses, you can get immunopathology. And that's shown here in the lung, where you can see you get a lot of lung infiltrates. This is occurring after about 6 days after challenge, so a little bit later. If you modulate that T cell response, and this is an inhibitor of, this is actually an arthritis drug, CTLA-4 or abatacept, which blocks CD28 co-stimulation, then limits the activation and proliferation of T cells, you will again limit immunopathology and cause that response to be a lot more productive. So you have both protection and immunopathology that is mediated again in the local site in the lung. So then how do we study this and figure out what's going on to a new infection? So in terms of, so we have to consider all the different immune cells that are in the lung. And this is done and we know this to be the case for both mouse and humans. So in human lungs, there's a lot of lung tissue resident memory cells here. So you have both CD4 tissue resident that are localized around the airways, as well as CD8 tissue resident memory. Now, they both express CD69. CD8 tissue resident memory are also CD103. A lot of them are CD103 positive. So they express that integrin that will mediate its interaction with epithelial cells, and then in doing so, they're actually they can be also in the airway. You also have large populations of tissue-resident macrophages in the alveolar space, as well as in bronchial macrophages, and you're also, again, the lung is highly vascularized, so you also have immune cells coming in from the vasculature, and that can be T cells. It could also be B cells and K cells and dendritic cells. So you have both these lung resident immune cells as well as immune cells that are coming in from circulation. So how do you follow then, and how do you study this in humans? How do you study what's going on in the lung and whether that response is productive, protective, or is causing immunopathology? So we've been doing this for a long time now in cohorts of patients that are in the pediatric ICU. So it's well known that before coronavirus, the most vulnerable of our population to respiratory viruses, to all different respiratory viruses, is really infants and very young children, and particularly to viruses such as respiratory syncytial virus, or RSV. So many babies, practically all babies, will get RSV, and most will recover, but some get a very severe disease and need supportive care and need to be intubated or need external oxygen support. And so what we've done is we've enrolled these patients and we get these respiratory samples. So either when they're going to clear out the endotracheal tube here as part of clinical care, it's washed out with saline to clear out that tube a couple of times a day. And so we've collected that wash and are looking at immune cells in that sample. If they have non-invasive respiratory sampling, we collect a nasal wash, and we found that it's actually fairly comparable, the type of sampling, whether it's upper respiratory or lower respiratory. And so far we've collected samples from over 150 subjects. Most of these has RSV, but also Rhino enterovirus, human metapneumovirus, and also seasonal coronaviruses. So about 15 to 20% of seasonal respiratory illnesses are caused by seasonal coronaviruses and other viruses. And we've collected these two samples as well as blood. And what you can see is that the airway sample is different from that in the blood. So the blood, and this is in infants, most of the T cells in blood are CD4 cells, whereas in the airway, you're getting both CD4 and significant fractions of CD8 cells. And you can follow these patients longitudinally because you're getting these samples every day as they're hospitalized and intubated. And what we found was that the samples from the infected patients, you had this sort of increase in CD8 cells increase in the frequency and the ratio of CD8 to CD4 cells in the airway that reached a peak and then resolved. So it's very similar if you think of sort of the dynamics of a T cell response to a respiratory virus that we're actually able to follow in humans. And then in the uninfected, these would be respiratory samples from infants that were intubated for non-infectious causes like cardiac surgery. and that we were able then to do correlates so that we were able to match components of what we were measuring in the respiratory samples with the clinical course of disease. And what we found, and these were done by Tom Connors, a pediatric critical care intensivist and scientist in the lab, and he found that the increase in the ratio of CD8 to CD4 correlated with lung injury and the pediatric definition of ARDS in the infected patients, and also correlated with increased viral clearance. So the higher the CD8 cells, the lower the viral titer. And so again, this is the nature of this overexuberant immune response. So the CD8s were coming in to clear the virus, but at the same time causing tissue damage. And that's important in terms of diagnosing and as an indicator for lung damage. So how do we then understand this and take these approaches to understand what's going on in COVID disease? So here is a diagram from a very nice and recent review article from Kanta Sabaru in Immunity, where looking at the sort of the different stages of COVID disease, either a mild disease or a severe disease, and what might be happening in terms of innate immune response and adaptive immune response. And then how did and of course, humoral immunity plays into this. There's been a lot of interest in when these patients make antibody, what type of antibody they make, and how can we distinguish these sort of immune response with the course of disease. And this is important for understanding in terms of immunomodulators. How do we define what is protective, what is pathological in COVID disease? So what we've done here at Columbia, and this was all set up during, as you know, New York City was the epicenter for the pandemic just a few weeks ago. Although we've really driven the cases down quite low. We've had a lot of success in doing that. But it's been 3 months. It's basically been 3 months, 100 days since this all happened. So the question we're asking is, what are the dynamics of the respiratory and systemic immune response in SARS-CoV-2 infection? And what are the immune correlates with lung injury or other clinical manifestations of disease severity, Asian risk factors? So we have an IRB to obtain respiratory samples, and this is our clinical collaborators are Matthew Baldwin in the ICU, in the adult ICU, and Thomas Connor in the pediatric ICU. We've obtained respiratory samples, so these are all endotracheal tube washes and blood daily from intubated COVID patients in the medical ICU and PICU. And in the height of this pandemic, the New York Presbyterian Hospital ICUs were literally all over the hospital. So they had their own ICU units. They also had most of the operating rooms were set up as pop-up ICUs. They had taken a lot of the hospital and really converted many units to ICUs. So this is from all over the hospital. And so what we're doing is we're assessing the immune cell composition by high-dimensional flow cytometry, and I'll explain that in a second, looking at their serum antibodies. And also we will be analyzing cytokines and chemokines both in the airway, aspirate supernatants and the serum, and doing transcriptomics. And the goal is to correlate immune parameters with clinical measures of severe severity, lung pathology, ARDS. And then look at these parameters as age, sex, and maybe different treatments. And this is all done by sort of the COVID team in my lab. These individuals, these are postdocs, Peter Chavez, Stephen Wells, Joshua Gray, Pranay Dogra, and others who have really worked tirelessly and in teams to collect these samples and do the assays and analyze them. So what I'm going to present to you now is really work in progress. And, we're seeing all these things on preprints. This is not yet a preprint. This is preprint. And because we're still analyzing, going through all these patients, so I'm just showing you sort of a preliminary analysis. So here are all the patients that we have enrolled and that we've got samples from. And so you can see that the age range is quite broad. It's 14 is the youngest and 84 is the oldest, and we have many ages in between. Most of these are male. Okay. We have most of these we have between about 7 days is sort of our average. There's one that we got 10 days on, but most of these we're doing for a week. We found doing it for more than a week ended up just being too much. And we were getting these nice trajectories from the week, from sampling for successive days within the week. And here are the outcomes that we know so far. So many of these patients are still sort of either intubated or in progress, or they're still recovering from the disease. A few have died. the lethality for severe diseases is about 30%, 30 to 40%, depending on where in the height of the pandemic. Some have recovered. Some have been discharged. When I have trach here, that means they have a tracheal tube. So they're still sustaining damage. And again, most of these is just sort of this is an active follow-up. Some have undergone to rehab. So what I'm going to show you first are the interim results. I'll show you the interim results from 7 patients. And so I'll start with the serum antibody responses. So we're still just kind of going through all this. That's why I don't have the full data set yet. So this will be the anti-spike protein. So that's been a lot of focus because of, as I mentioned before, that spike protein is what binds the cellular receptor. So antibodies that bind to the spike can potentially block infectivity because it's blocking to the binding site of where the cellular receptor. And those would be neutralizing antibodies. And there a lot of there's a lot of emphasis on this. And this is, so we did this neutralizing antibody with the Matteo Perotto and Anna Moscona lab, and this is done with a pseudovirus assay. And then I'll show you some cellular analysis in the longitudinal paired airway and blood samples and how we're starting to identify these dynamics and define them. So what about the neutralizing antibodies? Well, just to show you, the long and short is that all the patients that we've measured so far develop high titers of anti-S neutralizing antibodies. So what I'm showing you here are just individual subjects, and this is either the ELISA titer, and the blue is the first day, and the red is the last day. So you can see that some patients seroconverted and developed antibodies during the course of the hospitalization, whereas others already had high serum antibody. And you can see that for the last sample of all of these, they all do generate neutralizing antibodies. And interestingly, this patient here that had the highest titer in neutralizing antibody was a patient who died. And this was all done, again, by Stuart Weisberg in collaboration with Matteo Perotto and Anna Moscona's lab. What about so that they all have a very robust immune response in terms of their antibody response? But what about the cellular data? So the cellular data, we decided to go high dimensional here to get as much as we can, and because we want to look at everything, innate cells. And I mean, even though my focus is T cells, we wanted to look at everything. So this is our aurora spectral flow cytometry panel, and it's basically to cover all the major lineages: neutrophils, macrophage monocytes, T cells, B cells, NK cells, innate lymphoid cells, it's like gamma delta cells. And then, of course, we went and we have all these markers to define all different types of T cell subsets, including tissue resident memory cells, TRM, including regulatory T cells, effector T cells, CD8, CD4. And this is all done in the Aurora spectral flow cytometer. So we have now 150 samples that we're going through with all of these different high dimensional profiles. And that's what I'm showing you from 7. So from 7 patients, because this is high dimensional profiling and we have 37 or more parameters for each sample for both airway and blood. And remember, we have this for 7 days on most of these patients. So it's a lot of samples and kind of a deep dive into each patient. So we're using these sort of dimensional reduction approaches to visualize these data. So this is basically all of the airway samples and all of the blood samples from 7 patients for Donna for all their launching samples. Now, I will tell you, most of the cells, most of the CD45 positive cells are neutrophils, both in the airway and the blood. There's 90% of the leukocytes in the blood are neutrophils, and 95% or more are neutrophils in the airway. So we've gated out the neutrophils in order to study the dynamics of the other innate and adaptive cells. So you can see that, and this is a major lineage. So we set 5 major lineages: T cells, monocytes and macrophages, B cells, and NK cells. And you can see that between airway and blood, there's differences. So this is a UMAP projection. So it's a way to put in 2 dimensions, multi-dimensional data. So you can see that this part of the T cell here are missing from the blood, and I'll tell you right there, they are TRM, they are tissue resident memory cell, and then this macrophage cluster is here. This kind of part that's a macrophage is also missing from the blood. So these, again, the tissue is going to be different than blood. Blood has more B cells and has some more NK cells as well. But so this is sort of the average. And then if we can look at the average, and again, this is done by Pranay Dogra, did this pipeline in collaboration with other people from my group. So you can see that if you look at the longitudinal of all of the patients that we've that we've looked at, that you can see that there are, again, these compositional differences. So most of the cells are these myeloid monocyte macrophages across the board in airway and the blood, but the proportions of these differ. So the T cells are also a significant fraction in the airway, but they're a higher percentage in the blood. You also do not have significant B cells or many NK cells in the airway across the course of disease, but you do have, of course, good fractions of B cells in the blood and also significant fractions of NK cells and other cells. But again, this is the average of 7 patients. So we really need to look, this is humans, at every individual patient to understand what's going on. So what we've done in looking at this, and of course there's a lot of variability, but we can see and we can start to identify some trends. So the trends that we've identified, and we've put these into 2 groups, and this is all based on their airway profile, their longitudinal airway profile. So this group 1, or is when you have this sort of predominance and high predominance of monocytes, macrophages across the course of disease, way over T cells in these patients. And you can see that doesn't always reflect what's going on in the blood, but sometimes it does. So here's this middle, this COV OE10 also had this predominance of macrophages in the blood. But you can see this more dynamic appearance of T cells and some B cells in these patients. But interestingly, all 3 of these patients did not do very well. 1 died, 1 still has a trach tube, and 1 is also not doing very well. And then the second group that we found was a group when at some point during the course of us following this course of disease, that T cells predominated in the airway at some point. So that's sort of the second major classification. And you can see in these 3 patients, the blue line here are T cells, and they sort of overtake macrophages at different points, at different time points. And here in this individual, which is a younger individual, they predominate throughout the course of disease. And again, the dynamics in blood also sometimes reflected that in airway and sometimes didn't. But interestingly, and also in there was kind of peak in B cells in these 3 patients as well, kind of early on. Now, interestingly, and again, this is a small cohort, so please, this is just in our very small cohort, this group of patients did better. 2 of them fully recovered, and then 1 hasn't. One has been discharged, but has a trach tube. So just something to consider as we go on and move on to analyze more of these patients. Now, in terms of the T cells, again, I'm not going to show you longitudinal data. I'm just showing you one snapshot. Again, looking at these UMAPs, now drilling down, only looking at T cell populations. And what you can see, again, all of the airway and all of the blood samples from these 7 patients longitudinally, that you can see some major compositional changes in the airway compared to blood. And particularly these clusters, which I'm telling you based on the markers, are TRMs. So they're in the airway, CD8 and CD4 TRMs, and they're simply missing, of course, from the blood. There's also some interesting dynamics with some tissue Treg populations also going on in the airway, and that you don't really see so much in the blood. So we're very interested in trying to establish correlations with whether the TRM or the Tregs are correlating with any outcome. And then interestingly, you have more naive, of course, of T cells, CD8 and CD4 in the blood than you do in the airway. So we're going on and now doing the same type of longitudinal assays with the T cells. So just to summarize what I've told you and just sort of giving you kind of a snapshot of what we're finding out with these patients is, first, that local respiratory environment is important for immunity and immunopathology to respiratory viruses, and that in COVID-19 severe disease and is characterized by a robust immune response, they generate neutralizing antibodies. And we're certainly not the only ones to have found this with ICU patients with COVID, that others have also found that they're generating neutralizing antibodies and generating a robust immune response. So we found that respiratory immune responses are dynamic in severe COVID with significant populations of T cells and macrophages and monocytes, and that the predominance of myeloid cells in the airways throughout is associated with worse outcome in our very small cohort again, whereas increase in T cells, it seems to be associated with some recovered patients. And so the airway cells contain TRM and tissue Tregs, which may influence local immune response. So right now, what we're doing is, again, going through and analyzing all of these samples, determining whether immune cell changes correlate with protection or immunopathology by comparison with viral load and these clinical parameters that we've also, with Matt Baldwin and his colleagues, are assembling. So I just want to thank everybody in the lab who participated in this project, our collaborators in the ICUs and these nursing staff for these samples and clinical coordinators. And thank you all for listening. I'll be happy to take questions.
Really exciting and insightful talk. There are lots of questions actually that came through, and I'll try and go through some of them. And I've tried to group them in their relatedness so that you can cover a topic at one go. So, I mean, to start with, there have been a number of questions regarding the 1% asymptomatic individuals that you referred to in your first slide. And I'll ask you 3 questions related to that. The first one is, are we really saying these are asymptomatic patients, or we're failing to detect the classical symptoms simply because they're presenting with other symptoms that are not the classical ones for COVID? And related to that, then, is what proportion of the asymptomatic individuals will progress to active disease COVID, and what triggers that progression, for example? And if you isolate the virus from an asymptomatic individual compared to a symptomatic individual, is the virus sequence similar, or are there differences at all?
Yeah, so a lot of questions. So the asymptomatic. So we don't know why these people are asymptomatic. I think could be a few things. One is that just the virus, and there's been a lot of a lot of proposals and ideas of why people are asymptomatic. Is it just not infecting them, so they don't get a productive infection? And there's been a lot of children and young people are asymptomatic, and so there's been a suggestion of maybe they don't have as high a level of the receptor on the epithelial cells, of the ACE2 receptor, and that we just don't know that yet. Or it could be that the innate immune response is controlling that replication, or that they have pre-existing immunity somehow. If you have tissue-resident memory cells, and they will protect you from succumbing and appearing and getting those illnesses. So you could have pre-existing immunity that is really taking care of that. And maybe maybe there is cross-reactive coronavirus to T-cell immunity to coronaviruses, and that maybe kids have it more because they've recently seen coronavirus. So we don't know why. So it could be either pre-existing immunity or not. But I don't think the virus sequence would be different. I think people have shown that the virus sequence, there's not so many variations in coronavirus. Now, how it progresses to be symptomatic versus asymptomatic, again, that's probably due to the productive infection, both the productive infection in the cells, as well as the type of immune response. So if there's some delay in innate immunity, or then you're going to, again, get that symptomatic infection. Okay.
That's great. I mean, there were several questions as well to do with TRMs. And you've partly actually answered one of the questions, which was relating to if you've had previous exposure to coronavirus infection, not necessarily SARS-CoV-2, and developed TRMs, would those be protective against SARS-CoV-2, for example? And I guess you highlighted some of that response in the earlier response.
Yeah, I mean, so I think, certainly with influenza, if you have TRM to one strain of flu, and that tends to be cross-reactive, they will mediate a very good protection to a cross-strain, mediate cross-strain protection, at least in mouse models. So one can expect that if you will generate TRM to a previous coronavirus, and there's some shared internal epitopes, that you could potentially have cross-reactive TRMs to SARS-CoV-2. Now, the question is how long they last, how durable they are, and that we don't know.
Okay. There were a couple of questions relating to interferons in general. And again, in your earlier slides, you mentioned that the type 1 interferons are involved reasonably earlier on, I think, in the immune response to SARS-CoV-2. But there was a question which suggested that probably, is that response actually early enough, or is it delayed? and what happens to other interferons, for example, gamma interferon and others?
Yeah, so I mean, there's been experiments from people doing innate immunity, particularly at Mount Sinai, Hanover, and others, where they found that the type 1 interferon response is actually delayed or is dysregulated to SARS-CoV-2. And what happens is that just leads to this dysregulation of the pro-inflammatory cytokines and chemokines that are produced by monocytes and recruit neutrophils. So you get this dysregulated inflammatory response, and so everything's just a little bit dysregulated, and that will also cause kind of dysregulated, maybe induction of adaptive immunity, and that also can lead to immunopathology. So certainly elevated levels of IL-6 are correlated with worse outcome in these patients. And that's been done also here at Presbyterian. They found that as well. And so they've been treating with tocilizumab, which is that anti-IL-6 receptor blockade. Again, there's not enough, large-scale clinical trials to know whether that's effective or not. But certainly the delay and the sort of dysregulated, and that the virus doesn't promote a great type 1 response, is leading to this kind of altered or maybe even prolonged illness, as well as a prolonged induction or delayed induction of immunity.
Okay. There was a question asking on, in your earlier graph, you showed that the viral load actually goes down before you get the CD4 and CD8 or virus-specific CD4 and CD8 responses. The question, I think, is saying what really controls that virus earlier on? Is the innate immune response good enough to control it to that level?
Just so there's no misunderstanding, it's the peak of the T cell response or the antibody response always is reached after the virus is cleared. And that's not the case just with respiratory viruses. It's the case with like LCMV, with the lymphocytic choroid meningitis virus, which is sort of the model for a lot of antiviral immunity studies in mice. No, the T cell response it starts to be initiated and will go to the lungs. It's just you see the peak of it after the virus is cleared. But it's those T cells, it's T cell immunity, and it's the antibodies that are sort of mopping up whatever the T cells doesn't get. That's absolutely essential for that viral clearance to get rid of the virus. Absolutely.
Okay.
Yeah.
And a follow-up question to that was that is the virus really cleared in 7 days or so, both in the murine model, but more importantly in human disease as well?
Yeah, so we don't have the viral load on all of these patients yet. So I don't know in our samples whether we have that. I mean, there's other studies that are out there where they looked at viral clearance, and it is delayed. So in these ICU patients, you'll see the virus, and then eventually it is cleared. So I know that they've done, these viral load data here, and it is delayed. So it looks like the SARS-CoV-2, it takes a while to clear the virus. It could take weeks.
Okay.
Whereas the influenza, it's much, much quicker.
Right. And in patients who have significant lung pathology, from your studies, do you think that's mediated by a combination of innate and adaptive immune responses, or one arm of the immune response predominates in that lung pathology?
So it's very interesting, and it's certainly something we're really trying to figure out. So in those two kind of classifications where we looked at the airway and we saw that the macrophages predominated in one class of patients, and that the T cells kind of got in there and were able to, at least at certain points, sort of overtake, I think that might be a key. So if you're only having like a lot of macrophages and the T cells are not able to effectively do their job, then you're going to get this prolonged dysregulation and a lot of inflammation that might be inhibiting a productive response. And of course, that's going to lead to ARDS and a lot of lung damage. And that damage could be from these cytokines. Whereas if you can get some T cells in there which are targeted, which are going to go after those virally infected cells, that at least it lets in that window of hope. Now, you don't want too many of them going in, but having enough of them go in seems to be key. And that's going to at least control that sort of dysregulated. So if you have just a lot of infections and you have mostly these innate cells and not enough T cells to kind of clear out those epithelial cells, then you might have this prolonged dysregulation. And it's just sort of interesting. In one of the patients who did quite poor, who died actually, the T cells are not there in the airway. And at the very end, they start to increase in the blood, but they're not yet in the airway. You kind of think, were they trying to, get that T cell response in there? Yeah. Whereas some of the younger patients are able to make really robust T cell response that get into the lung. So we're going to have to look at this more. That's just sort of a hypothesis based on the limited analysis, but that's what I would propose. Yeah.
Okay. That's really interesting. So there's a question regarding rendezvous. Does it have any impact at all on lung pathology from your knowledge and from studies?
So I think, we're going to have to look at which patients got rendezvous. So what happened is it's sort of like in the middle of all of this, that study came out that it was showing some good effects. I mean, a lot of these patients in the ICU, they did throw a lot at them, just sort of trying to get something, it's just that there was this sort of this helpless period, I think, until they learned, how to manage these patients. So I don't know. I think if it's going to clear the virus, it will clear some inflammation, but I think it depends in the timing of disease for a particular patient. So if that patient has been sick for a while, that the antivirals, just like with flu, it's going to be too late, that the damage and those pathways of damage have already they're already underway, and the antiviral is not going to affect it. So that's when you want to go more to the immunomodulator. So I think what's going to happen is just like Tamiflu, you need it early. It'll be the same thing with rendezvous. Okay.
I know in New York you've seen a lot of patients, but I don't know how many of these you've followed up post-COVID. But there was a question asking, are these patients at increased risk of secondary bacterial infections even after recovering from COVID, for example?
Yeah, so I mean, certainly during the illness. So we have excluded those patients who develop secondary, really severe bacterial pneumonia, because then we were a little worried that we'd just be looking at bacterial pneumonia. So we have excluded those patients, but it was a significant frequency of patients, especially older patients. So a lot of these patients are from nursing homes, some of the really older ones, like an 84-year-old and some others. So they also are at risk. Now, later, once they've recovered, I think that's a really big open question. So how much lung damage is sustained from people who have had severe COVID and then recover? I think this is a really important question. It's also for people who aren't hospitalized, who were at home with a severe illness.
Okay. A couple more questions then. Do you think an intranasal vaccine for SARS-CoV-2 would be better at generating TRMs and therefore offer better protection in future?
Yes, I do. I think that, I know there's a lot of focus on the neutralizing antibody, and all the companies are just sort of putting everything in this. And I think that will be great. I think any vaccine, whether it provides sterilizing immunity or not, is going to help, and people should have it, even if it just protects from getting a less severe disease. But certainly intranasal at the site. So I took out that slide; it was just too long. But we've certainly shown, and others have shown, in other respiratory viruses like Kingston Mills for pertussis, if you go intranasal with some sort of live attenuated, you get TRM. It's the only way you get TRM in the lung. The systemic, the flu jabs, don't give you TRM, at least certainly not in the mouse, right? If you do the same thing analogous to the mouse, you don't get it. So you will get protective TRM with an intranasal live attenuated vaccine. So I would really push for that. I think we should. I think they are doing it. It just hasn't gotten as much press. So I hope we'll get more, because I think we do ultimately need these respiratory TRM.
So there was a follow-up question to that, actually, saying if that became a reality, if we had a vaccine either administered intranasally or otherwise, would patients who have recovered from COVID be eligible for such a vaccination program, or you'd think that we don't know yet, but you'd think that they've got sufficient immunity to protect them, for example?
I think that everybody should get it. I mean, there was actually is a really interesting study. It's a vaccine trial on the MERS camel vaccine trial. It's a paper in Scientific Reports, and I'm blanking on the authors right now. But it's a recent paper. It's in Scientific Reports. It's MERS. It's a camel study. So where they looked at the vaccination for the camels, and they did camels. It was very similar, it was like a herd study. So they had camels that had previously gotten MERS, but recovered, so were seropositive, and then the younger naive camels that hadn't seen MERS. And it turned out that the camels who responded the best to like an anti-spike protein vaccine, so it's very similar to the vaccines, like this is an adenovirus vector with the spike protein. The camels that were seropositive actually responded the best to the vaccine, so they had an increase in their titers, and they did better than the naive camels. Naive camels, some of them seroconverted, but it was the camels that were previously had the illness that did the best, suggesting that you may have to be re-exposed again and again to really, augment that immunity. So I would say that, yes, vaccinate everybody, whether you've had the illness or not, because it may be additive.
Donna, thank you so much for a very interesting talk. I've learned a lot, and I'm sure that all participants have learned a lot as well. It's been really informative and very timely as well. And to all participants, I'd like to thank you for your time, for staying on to listen to this talk. My name is Henry Mwandumba. I'm the president of the Federation of African Immunological Societies, and I've been your moderator today. And thank you so much. And we'll see you again at the next webinar, which is on the 15th of June, which is only Monday next week. Thank you, Donna, and thank you, everybody.
Thank you, and thanks, everyone, for their comments. Thank you.